Now we are going to switch to computer science. The two presenters are Albert Toth and James Andress, both of them from Oak Ridge High School in Oak Ridge, Tennessee.
Hello, ladies and gentlemen. My name is AJ Toth, and this is my partner, Jim Andress. Our project is entitled Creating a Higher Efficiency Machine Learning Algorithm for the Development of Cancer Treatment Drugs. The goal of our project is to save time and money in cancer research by creating an algorithm which accurately predicts target protein mutations. We combine stochastic discrimination and active learning in a novel approach, and our algorithm has applications in many fields. Two types of mutations must occur in order for cancer to grow and develop. First, there must be a mutation in the cell growth pathway. And second, there must be a mutation in the programmed cell death function, or apoptosis. Now, in this diagram, we show an accumulation of mutations in the cell growth before there's a malignant tumor. If at any point during the sequence the mutated cells had been killed through programmed cell death, or apoptosis, the sequence would not have been allowed to continue and cancer would not have grown. The p53 protein regulates the cell cycle by responding to cell stress. When there's stress in the cell, the protein binds with the DNA, either to repair the mutated sequence or to initiate apoptosis when necessary. However, in approximately 50% of all cases of human cancer, the p53 protein is mutated in such a way that it cannot perform its function. When the protein is mutated, it can't bind with the DNA, and the cancer is allowed to grow and spread. Research has shown, however, that some second mutations of the p53 protein can actually reactivate its functionality. Research seeks to find the reactivated p53 proteins as they can halt the spread of cancer and even suppress well-developed tumors. In our project, we use the p53 data set from the University of California at Irvine Data Repository. It contains 32,000 examples of some of these second-site mutations of the p53 protein, each with 5,409 different features. The first of these features is the label of active if it reactivates the protein's functionality, and inactive if it does not. Other features include the mutation location in the protein surface features, distance measurements, and estimates of protein thermostability. However, in testing our algorithm, we focused on a small 261 example set that has been previously studied for comparison purposes.
In order to analyze the p53 mutations, we combined two machine learning techniques, the first of which was stochastic discrimination. And stochastic discrimination works on the idea that rather than generating a single high-accuracy model of the data, it's more efficient to generate thousands of weak classifiers and then combine them into your final model at the end of the process. For example, given the set of blue points and red points, the goal of the algorithm would be to determine the label for the previously unseen gray point. Now notice that this is a two-dimensional example, while we're actually working in over 5,000 dimensions. And in our application, the labels of blue and red would correspond to the active and the inactive mutations. In order to classify the gray point, the algorithm selects a random section of the feature space, which is known as the subset S. And as you can see, the majority of the points within the subset are blue. Therefore, the algorithm makes the generalization that all the points in the subset are blue and all the other points are red. This classification is done with the equation shown where M is the unlabeled point, S is the subset, and C1 and C2 are the two classes of points. Notice that this classifier isn't very accurate, as it only predicts the correct label for 52% of the points. Therefore, we generate thousands of these subsets. And as you can see, the majority classify the gray point as blue, although occasionally there is a subset that classifies it as a red point. However, after generating thousands of these subsets and seeing that the majority classify the gray point as blue, we're confident that it is in fact a member of the blue class.
One of the problems of machine learning is that it requires a large labeled data set in order to run. The labels for this data can be expensive and time-consuming to obtain. So to solve this problem, we implemented an active learning scheme in our algorithm. Active learning works by selectively choosing new examples to be labeled to quickly build an accurate classifier using as few expensive labels as possible. So once again, in our two-dimensional two-class example, we have red points, blue points, and unlabeled gray points. The active learning algorithm begins by separating the labeled data for a training set to build a model. The model is then applied to the unlabeled test data. From the test data, the algorithm chooses the new examples which will most improve the model by being labeled. In this case, it's the 4 in the center, as they will most define the boundary between the two classes of points. The algorithm then labels these new examples, adds them back to the training set, and then the process repeats. By doing this, we can quickly build an accurate classifier and use as few expensive labeled examples as possible.
Our approach combines stochastic discrimination and active learning in a novel way. To begin, we started by randomly selecting 25 examples to use as the labeled training data and use the other 236 examples as the unlabeled test set. From there, we built a stochastic discrimination model on the training data, then applied the model to the test set. And this evaluation allowed us to select two groups of points. First were the likely actives, those mutations most likely to reactivate p53 functionality. And second were the boundary points, those points which the classifier is least confident about the label. We then selected and labeled a combination of the likely actives and the boundary points, moving them from the test data to the training set. And then finally, this process of model, evaluate, and update was repeated until the halfway point was reached. And the halfway point is a measurement of efficiency in machine learning algorithms and is equal to the proportion of the unlabeled data, which must be labeled to find half of the positive examples. This means that a low halfway point corresponds to a high efficiency model.
Several parameters had to be optimized in order to achieve the highest efficiency in our algorithm. First, we had to choose the number of new examples to label during each round of active learning. The number of examples labeled must be small enough to allow the model to frequently refresh itself, but it also must be large enough to add sufficient information during each round of active learning. Our test showed that the number of new examples labeled, alpha, was optimal at 3. Next, we had to set the threshold between choosing likely active points and boundary points. The threshold is a y value which can be thought of as the likelihood of a point being active. So a low value of the threshold will focus mostly on the likely active points and less on the boundary points. But a high value of the threshold will focus mostly on the boundary points and less on the likely active points. Our test showed that the threshold, tau, was optimal at 0.65. In other words, in order for a likely active point to be considered to be labeled, it had to have a likelihood of activity above 65%. Both of these graphs plot the same data, but this graph is alpha with different values of tau, and this graph is tau with different values of alpha. Each is plotted with its respective halfway point value. And since the lowest halfway point is desirable, the optimal values of alpha and tau are seen when alpha equals 3 and tau equals 0.65.
In addition to alpha and tau, we also had to optimize an ignore factor parameter, eta, which was used to compensate for the fact that we have a high dimensionality data set but few examples to test with. We found that a value of 0.999, in other words, ignoring 99.9% of the features, was optimal. And it seems counterintuitive that as you ignore more information, your results improve. But what's actually happening is that each subset is getting its own unique perspective on the data. And because the specific features which are ignored change, no information is lost in the process. We found that as eta, the ignore factor, increased, the halfway points dropped, meaning that our models were generated more efficiently. Finally, we had to choose t, the number of subsets for each stochastic discrimination model. Obviously, generating more subsets takes a longer time, but a large enough number of subsets is necessary to counteract the random effects of the stochastic discrimination process. We found that a value of 5,000 subsets per model was optimal, as it produced consistent results with little deviation for multiple runs of the same starting parameters, but was also generated quickly, even on the laptop computers we used for our testing. This chart shows various active learning techniques, as well as their halfway point values, all generated using the same data. You can see that our technique, stochastic discrimination with active learning, highlighted in blue, has the lowest halfway point, and therefore the highest efficiency. An explanation for this is that the other techniques shown are MIP, standing for most informative positive. This means that these techniques, given the choice between labeling a likely active mutation and inactive mutation, will always choose to label the active. However, we recognize that sometimes labeling the inactive mutations does improve the model, and therefore we chose to label a combination of the likely actives and the boundary points, getting the best of both worlds from our models.
If our algorithm were applied in a true experimental setting, the process would be as follows. We begin by testing a small number of examples for a labeled training set. Usually testing an example involves running it through a laboratory to determine the class label as well as all the features associated with it. This data would then be recorded in a data set, and the algorithm would run. The algorithm would then yield the results, which identify new examples that will most improve the model by being labeled. We'll then test these new examples as identified by the algorithm by running them through the laboratory to determine class label and features, add that new data to the data set, and repeat the process. The cost of labeling each P53 mutation in the laboratory is approximately $40 in yeast-based assay materials, $300 in labor, and requires approximately 10 days worth of time. So to reach the halfway point in our small 261 example set will cost approximately $40,000 by simply randomly labeling new mutations. However, using our algorithm would only cost $22,500. This is savings of 44%. If these savings were applied to all cancer research everywhere, the total savings in time and money would be huge. And in addition, the increased efficiency could lead to quicker discovery of effective cancer treatment drugs.
In conclusion, we successfully combined stochastic discrimination and active learning in a new technique with a higher efficiency than previously explored methods. This higher efficiency would lead to savings in time and money in cancer treatment research and is therefore the first step in the development of a viable cancer treatment drug. In the future, we'd like to add to our algorithm to further explore the feature space and to identify the features which most indicate a particular class. Given the success we've had in the cancer treatment field, we'd like to apply our technique in other areas, such as biology, material science, or pharmaceutical development. Hopefully, we'd be able to achieve the same high-efficiency results that we've had in cancer treatment research. These are our references, and we'd like to thank these people and institutions, specifically the Siemens Foundation, College Board, and the George Washington University, as well as the audience and judges for their attention. Thank you.